Thanks. Right. So we switch the gear a little bit from RNA to DNA methylation. So DNA methylation is when a methyl group replaces a hydrogen on the cytosine, then forms what we call methylcytosine. So it's one of the two major epigenetic mechanisms. So this DNA methylation is known to play a very important role in gene silencing, activation, cell reprogramming, genomic imprinting, and tumor genesis, cancers, right? And now with this, of course, with the high throughput sequencing, we can now obtain the entire epigenome or methylome. I'm just going to use the two terms exchangeably here. Those sequence. And the first whole methylome was obtained in year 2008 for Arabidopsis, a model plant genome, and using the technology called bisulfite sequencing. So the converted sequence, the converted and amplified sequence were sent to, fragments were sent to the sequencer, and the reads, of course, were just aligned to the reference genome, just as other sequencing. So as a result, in addition to the DNA sequence, we also obtained the state of methylations at each cytosine site. Sometimes the sample is maybe consists of a mixture of epigenomes. How so? Because the methylation patterns sometimes may vary among different tissues or different cell types or cell types of different developmental stages. And in addition, more and more evidence have shown that the aberrant methylation is a generic feature of cancer genomes. So if we can understand these methylation patterns of cancer cells, then perhaps those can be used as diagnostic markers. And if you're really looking ahead, and you perhaps can even use them as therapies, as methylation changes as an epigenetic mechanism are reversible, unlike DNA mutations. But often the times, the cancer cells and the normal cells are mixed together, particularly in the early stage of cancers. So in order to make use of the DNA methylations of the cancer cells, in this kind of situations, first we need to untangle the mixture of these epigenomes. And another scenario is sometimes you do sequence the single cell type. And when you look at the features of the methylome from single cell type, there are also certain observations that backs the explanation. For instance, when they compare the methylome of human embryonic stem cell, H1, with the fibroblast cell, MR90, and MR90 overall is less methylated than H1. In particular, there are certain regions on the MR90 that are partially methylated. And this partial methylation is observed in 80% of the X chromosome, which is not unexpected because MR90 is cell line is from a female. And methylation is known to play an important role in X inactivation. What is unexpected is it's also observed in nearly 40% of autosomes. So immediate question is, what is the nature of this partial methylation? My allelic differences contribute to that. In order to answer that question, we first need to detect know how to detect allele-specific methylation patterns. So these two scenarios I just described is really one type of problem. That is, you have the reads from a heterogeneous sample that mapped to a reference genome. Except, of course, we don't know which read is from which one, what we call epitype. So what we would like to do is, of course, to reconstruct it, to know which, to assign a read to a particular epitype, and hopefully to reconstruct the original one. So At this point, some of you in the audience might ask, wait a second, isn't just like the variant calling problem at the local level, or if you really need to put them all together, that's the haplotyping at a global level. After all, DNA methylation, methylcytosine, is sometimes referred to as the fifth nucleotide, right? With a very important difference. DNA methylation is a lot more dynamic than nucleotides. They often appear as statistical events. Consequently, your methylation rate at each site can really range from anywhere from 0 to 100%. This is different from SNPs. And so in order to assign the methylcytosine along with its reads to its respective epitype, or in this little example, let's see allele, is really a more complex problem. But sometimes we might be lucky if in the near neighborhood we also have some SNPs very close to the methylcytosines, such that they actually reside on the same reads. Then the problem becomes trivial, right? Because you can just use SNPs to assign the alleles in this example. Then immediately you can say, oh, so one allele has 50% methylation while the other is unmethylated. But what if we're unlucky that we do not have such SNPs to inform us? Then we're back to our original problem, which is unfortunately most of the case. Because DNA methylation happens at a lot higher frequency than the SNPs. What I'm showing here is just example for the human. It's known to have around 3 million SNPs, while for the stem cells, H1 cell line, 62 million methylcytosines have been detected. In the fibroblast cell line, MR90, 45 million. But of course, there's hope. If all you have are the single methylcytosines on each read, really we just don't have enough information to make any inference here. But if you have some neighboring methylcytosines residing on the same reads, then they can inform each other. As long as there's a sufficient difference between the different epitypes, then the hope is we can, in fact, separate them and tangle this mixture. So that in addition to what we are having right now, which is an averaged pattern of the methylation, we can also have the two individual methylation patterns. So this is basically the intuition. And from that, based on this intuition, we approach the problem using a mixture model. So the observation is just a set of reads, right? And then if we know the epitype frequency, and we can think of the reads are generated as follows. You draw an epitype from the population according to its frequency. And then we have a matrix of that represents the probability of methylation on each cytosine site as its column for each epitype as its rows. So one epitype is really just a row in this methylation matrix. Then a read is just, if you take a fragment from this epitype and send it to a sequencer, you end up with a read. And of course, the fragment on the epitype, what you have are the probabilities. And the read in the end is your observation. Since we only care about methylation, we can represent it as just a binary array, 1 being methylated, 0 being non-methylated. And then we can easily write out the probability of observing this read from this epitype. And of course, we don't really know which epitype this read is from, so we really have to count for all of them. You just sum across the epitypes. So this is the probability of observing a single read. And then if you just multiply across all the reads, then you get a probability of observing the entire set of the reads, or similarly log likelihood. And of course, we don't really know the methylation matrix, or in many cases, the epitope frequency. Those are exactly what we're trying to compute. So our goal here is really just to find this methylation probability matrix and the epitope frequency such that the log likelihood of observing the entire set of read is maximized. So this is essentially our model and our goal. And of course, this optimization can be solved using EM algorithm, expectation maximization algorithm. So in the end, what we obtain is estimated methylation levels at each cytosine on each epitype. And we'll also have membership of reads in probabilities, and of course, estimated allele frequency. So this is really the core of the method. And then we apply this model to the detection of allele-specific methylation. Primarily because at the time that's what we have data for the availability of data and possibility of validation. Excuse me. So this is the core of the model, and for pre-processing, we scan the entire methylome for partially methylated regions, because really there's not much point to look at the region without any methylation or with very highly methylation, because clearly they're not allele specific. And then once we obtain the estimated methylation levels, we send this through some classifier filters, and in the end, we announce, okay, those are predicted allele specific regions, or if they're not. So of course, we need to test our method. So we also generate a lot of simulation data, but for validation, we also use the synthetic methylome because we think this is really resembles a reality a lot closer than any simulation. What we have is we have two Arabidopsis root cells. They are all of single cell types, and we have the methylomes for these two cell types, and then we take all the reads from these two methylomes, we just mix them together, and that's our synthetic methylome. Then we pretend one cell type as allele A, the other cell type allele B. So first, we again just do some pre-processing, and if a region is partially methylated, we use some very simple criteria to label the region as whether cell-specific or not, or in the terms of synthetic methylome, whether it's allele-specific or not. We label these regions, then we put the labels away. We send the reads of all these regions through our model, and then we do some post-processing. We give a prediction, then we compare to our original label. So now let's look at some results. So this is one of the cell-specific methylated regions. The x-axis is just the index of methylcytosines, and the y-axis is methylation rate ranging from 0 to 1. Two colors represent two cells. And these are computed directly from the reads. So these are sequencing results, experimental results. This is what we treat as golden. And then this is the predicted methylation levels for this region. And you can see the pattern tracks rather well. And then this is an example of non-cell-specific methylated regions. Again, experimental. So this is the methylation levels really mixed up quite a bit between the two alleles. And this is the prediction. Again, the pattern tracks. Of course, the algorithm tends to exaggerate the separation a little bit, but not too much. So once we obtain all this individual methylation level, the methylation levels at all this individual size, we still want to look at the entire region try to characterize region, whether it's allele specific or not. So I should mention here, why do we care about the regions rather than the individual size? After all, when we look at the SNPs, we just look at the individual nucleotides. It's a general observation that the methylation level at individual cytosines does not have much of functional consequences. Rather, it's really the methylation levels across certain regions that plays a more important role functionally. So that's why we really focused more on the region rather than the individual size. So we would like to characterize those regions, but really what is allele-specific methylation is not a well-defined term. Unlike the SNPs, either you have it or don't have it. So we just use some, mostly we get some features based on intuition. And if you're eyeballing a region, the methylation levels, you can, basically we can say, yeah, this is allele specific, this is not. So we basically extract a few features from this predicted methylation levels and the read assignments, and we build an SVM classifier. Those are standard procedures through cross-validation, of course. And then we tested on about 100 pairs of samples that were set aside just for testing. The overall, this is the confusion matrix for the testing results, and the overall accuracy is not bad, 87.5%. So we have some confidence in our method. Then we went ahead to predict allele-specific methylation in real data, in a single methylome. We applied it to one of the two root cell methylomes we had. So there were about 450 some partially methylated regions. We send all those reads through our model. And then for the post-processing, for characterizing each regions, of course, we used the SVM classifier trend on the synthetic methylome. One of the root cells is just this. But you probably already realize this classifier really has limitations, mostly because in practice, a classifier trend on one organism cannot be used on another. And in a lot of cases, you may not even have enough data to train a classifier like this. So in addition to just relying on that, we come up with just a few more filters or statistical tests. And also, we're trying to be very conservative here. So one of the filters we use is what we call low methylation rate. Basically, look at whether one of the cluster has very low methylation level. And the other is what we call differential average methylation rate, basically contrast the overall average methylation level between the two alleles. And then based on this measure, we put the regions into three categories. One is what we call highly differentially methylated. The other is moderately differentially methylated. Of course, the third class is just similarly methylated. And then we take the intersection of all these measures just to reduce the false positive, because we're trying to predict the least specific regions. It's okay if we miss. So we came up with 18 such regions. Let's look at just one of them. So again, this is predicted the methylation levels, same graph as you saw before. And on the left side, those are just reads aligned. And again, the colors represents the leads, but those are assigned. We don't know the truth now. And each dot on this line is a methylcytosine. And at the corresponding positions, when there is no dot, it just means that site is unmethylated. So these are the actual reads. And the colors are assigned based on the probability of the membership, read membership. And this is the predicted methylation levels. As you can see, these are very different. The methylation levels are just one allele has nearly 0, the other is nearly 100%. All of these regions are like this. If we lower our criteria a little bit, we'll of course, we'll end up with a few more. If we look at one of such regions, the real difference is one of the leads the methylated alleles is not like 100% methylated in the entire region. The separation is still very, very good. Again, I should mention all these are predictions. And if we remove the most stringent filter, we'll end up with a lot more prediction, 170 some. And this is, again, one of such region. Again, you can see basically this is just a scale issue. The methylated allele has a much lower overall methylation level, not nearly 100%, whilst the non-methylated allele is not zero methylation. It has 20% on average here, 20% probably, yeah. So, and then, so of course, really, once one predict this, you will want to look at the functional consequence if you have the data. So then one should really focus on these two groups and using this as those are the candidates. Nonetheless, we just look at annotations of all these regions, and some of them overlap with genes on the gene bodies, and quite a few of them is at the upstream of a gene. So those would be the ones that one want to spend more time on to look at what the expression profiles. We didn't have the data at the point, but really that's what you want to look at it, because methylation on the promoter region is known to silence gene expression. So if you have allele-specific methylation near at the promoter region, then you want to see whether there is a corresponding differential allele-specific expression associated with that. All right. So unexpectedly, a lot of the regions just overlap. Is on the transposable element regions because we're looking at a plan, and demethylation it just plays a very important role in transposon silencing. So one thing we wanted to look at is the imprinted genes, because methylation has a role in gene imprinting. Unfortunately, the reported imprinting genes in Arabidopsis is very sporadic. It's just not many of them being reported. We did find one of our predicted region overlap with one of the few imprinting genes in some way. This gene was reported imprinted in the Arabidopsis seed. We don't really know whether it's imprinted in the root or not. And of course, we don't know is there any functional consequences here. So basically, that's what I have. To summarize, we've developed the computational model for resolving distinct epigenomes from a heterogeneous sample. And we apply this model to the detection of allele-specific methylation in absence of SNPs. We don't need the SNPs to inform us. So, of course, there are many directions one can go from here. I just want to mention that this is really just the beginning of decoding partial and differential methylations. And why do we care? Because this may account for differential expression, and this may act as marker for individual clones. And in tumor genesis, this may form basis for tracking stem cell growth and tumor expansion. Right. So this work was done while I was at UCSD and the Salk Institute. I have a number of people to acknowledge, and of course the funding agents, and then the travel fellowship. With that, I thank you, and I can take questions.
Hi. Very interesting work. I had a question. So I saw Andrew Smith's lab in PNAS had a very recent paper with a similar goal, and I was wondering if you've compared their method with your method, and do they give the same allele-specific predictions, or are there discrepancies?
No, you're talking about Andrew Smith's group?
Yes.
Yeah, actually, I just heard of it. No, they applied their method on human, so we really didn't compare the data. I think the method turns out to be similar, if I'm right.
Okay.
Yeah.
But nobody's applied these two methods to the same data set.
Actually, I wasn't aware that until the paper came out. Yeah.
Okay.
So we didn't compare. And the reason I worked on Arabidopsis, of course, one thing is I'm in that lab. The other is at the time the humans, because I was interested in the entire methylome. The humans are only you really only see very dense methylations in the CPG islands. And where elsewhere you just don't have enough methylations that will reside on the same reads, then you can't do much about them.
Okay, thank you.
Yeah.
Okay, can I have a question? Can you use methods deal if you don't know the number of epitides? Because now you assume that you
Yeah. I see your question. Right. Yeah. So here I assume I know it, right? Yeah. So that is not the problem that it's being solved here. Right. So of course, for Allele-specific methylation, since we know it's a diploid, the number is 2. And when you want to apply this model to say a mixture of cells, you don't know how many. You don't know the number of K, basically, right? So there are different ways one can do it. One is you could. That's probably what a lot of people are doing with this kind of clustering, basically. You try different Ks, you try different number of clusters, right? Your algorithm will always give you something, and then you develop some statistical test to choose the better one.
Okay.
That's one way of doing it. And we have actually another paper that using some combinatorial approach that will optimize to the minimum number of epitype, haplotype, or whatever. But again, it's you set up a computational goal first. Yeah, so it's, as I said, it's so really in practice, you will have to look at a few and really test which one best explains your data. Let's say even you have 10 cell types, that doesn't mean you have 10 epitypes. Right. All right. Thank you.